This lesson is an introduction into osmolarity and how water potential can be used to measure osmotic pressure. Let's start with the tale of blood and water. Human blood cells are found in an aqueous or water-based solution. What this means is the concentration of solute to water, both in the cell and the solution it's in, can vary at any time. For example, if I were to look at human blood under a microscope and there was the same amount of solute inside the cell and outside of it, this is what I would see. Healthy, happy red blood cells. They have their typical circular shape and they're functioning well. But what if I skew that concentration a little bit? What if I put these cells in a solution that has a greater concentration of solute? These cells at first will be fine, but as time goes on, because of that difference in osmotic pressure, you can see here the cells begin to shrivel. They begin to shrink and the cell membrane begins to infold. Interesting. What if I change the balance in a different direction? What if I put more solute inside the cell or put them in a solution that has less solute? This time, you can see the cells are actually getting bigger. They're starting to swell, and this continues until cells can no longer hold it and they burst. So what's happening here? Why is water moving in the first place? Why are these cells either shrinking or swelling and splitting instead of just staying the way they normally are in the first solution? This is an example of osmosis. Osmosis is a diffusion of water from a high concentration to a low concentration across a selectively or semi-permeable membrane. You can see in this animation here, we start with more water on the left-hand side of the cell, and then it moves to where there's less concentration of water, from a high to low concentration. And in this example, our selectively or semi-permeable membrane is the cell membrane. So let's look at an example of osmosis in action. Here I have set up a U-tube apparatus. On the left-hand side, I have a couple solute molecules and a good amount of water. On the right, I have more solute and less water. And these two sides are separated by a selectively permeable membrane. Take a moment, predict what you think the movement of water will be in this system and what the movement of solute will be. Let's see if your prediction was right. Interesting. So water moved to the right, and now you can see there's dramatically more water. The water on that side of the tube is moving up. But the solute, I have 1, 2, 3, 4, 5, 6 on the left side here, and 1, 2, 3, 4, 5, 6. Solute didn't move at all. Would you be able to explain this? Could you explain why the water moved to the right instead of to the left, and why the solute didn't move at all? Well, why did the water move? Osmosis, the diffusion of water from a high concentration to a low concentration. When we started this apparatus, there was more water on the left, so it moved to where there was less water on the right. And it looks like water was permeable, meaning it was able to move across the selectively permeable membrane. Solute, on the other hand, didn't move at all. Why? Well, this membrane selects against the solute. The solute particles are too large to move across the selectively permeable membrane. But notice this interesting circumstance. Water will raise higher, which is quite counterintuitive because of that osmotic pressure, because water will always move to where there's a lower concentration. So cells have an osmosis problem. Cells are surrounded by water and they're made up of water. So how is a cell going to be able to keep a perfect balance of water, having the same concentration outside as within? Turns out that the survival of a cell really does depend on its ability to balance its water intake and output. For humans, when we have cells that are put in fresh water, water's going to rush into our cells and cause them to get larger. Ideally, we would want a solution that has the same amount outside as inside. At that point, we're perfectly balanced. But we can also have the scales skew in the other direction if we're put in a saltwater solution. In a saltwater solution, there's more salt outside of us than within, and that causes water to leave the cell. Let's look at this example. A cell containing 10% of salt is placed in a beaker that contains 2% salt solution. So I have more salt in the cell than I do outside. How do you think osmosis will occur in this situation, and why? Well, water's going to move into the cell. Why? Osmosis. Remember, osmosis is the diffusion of water from a high concentration to a low concentration across a selectively permeable membrane. In this situation of a cell inside of a beaker, that 2% salt solution has 98% water, whereas the cell, being made of 10% salt, only has 90% water. So osmosis, water is going to move where it's greater in the solution in this example to where it's lesser inside the cell. When a cell is placed in this kind of a solution, we refer to it as a hypotonic solution. These are solutions that have a lower concentration of solute, conversely a higher concentration of water, than the cell itself. And when this occurs, water is going to move into the cell. When that happens to an animal cell, cells that don't have a cell wall, the cell is going to get bigger and bigger and bigger until it splits, or what we technically call lysing. That's what you saw with the red blood cells in distilled water. Distilled water has nothing in it but water, so it has a lower concentration of solute. In plant cells, however, plants have a cell wall. That wall, it has a lot of integrity and is able to maintain its structure and pressure in these situations. So if I put a plant cell in a hypotonic solution, it's fine. The cell wall will protect it from bursting. In fact, the cell will become more firm, which is ideal for plants. Think about freshwater organisms like this single-celled protist here called a paramecium. This organism is in freshwater, meaning that the environment it's in always has less solute than it does inside itself. Living things need salt and other particles in their solution to survive. Freshwater, on the other hand, very, very little. So how is something like a paramecium going to be able to overcome this problem of water constantly rushing inside of it? Well, they're just going to pump it out. They have a specially adapted vacuole called a contractile vacuole. What this vacuole will do, and you can see it in this animation here, is it'll fill up with that excess water, and then it'll get pumped and squirted out of the cell. So even though water's constantly rushing in, the paramecium can consistently pump it out to maintain the perfect balance of water. Now keep in mind, this requires ATP. Staying alive is expensive. Creatures like the paramecium have to consistently acquire energy to use it just to maintain their osmotic balance so that they don't swell and lyse. Here's another system I want us to look at. This time I have a 10% salt solution and a cell that contains 10% salt. What do you think is going to happen in this system? Well, this system's perfectly balanced. I have the same amount of solute outside as I do inside. Solutions like this are called isotonic, when there's no difference in the concentration of water between the cell and its environment. This is when the cell is in equilibrium. This was that first video of blood cells that I showed you. In this instance, there's no problem. It's a dynamic system. Water's going in, but the same amount of water's going out because the concentrations are the same, so the cell maintains balance and it is fine. Let's look at this scenario, however. What if I put that same cell with 10% salt in a solution of 30% salt? What do you think will happen now? Well, water's going to move out. In this kind of a solution called a hypertonic solution, this is a low concentration of water, but a high concentration of solute in the solution. Since there's a high concentration of solute in the solution, think of this like putting one of your cells in salt water. Osmosis is always going to move water from higher to greater. So since there's more in the cell, it'll move out. For animal cells, this will cause them to shrink and shrivel, like those blood cells that were starting to shrivel in on their sides. For plant cells, it's quite a unique situation. The cell wall is sturdy and it's not going to move, but the plasma membrane underneath the cell wall will actually detach and shrivel inside the cell. This is something we call plasmolysis. Plant cells can survive this scenario. It is not a death sentence, and it is again because of that cell wall keeping everything intact. So in any of these solutions, a cell's survival does depend on the balancing act on how it can interact with different concentrations of solute in its solution. If it's in a hypotonic solution where there is more solute inside the cell, it needs to pump that water out or it's going to lyse. If it's in a hypertonic solution where there's more solute outside the cell, water's going to rapidly leave. It's going to need to figure out how to pump more water in or swim to a more hospitable environment. But when everything's in balance and I have the same concentration outside as within, I'm in an isotonic solution and the cell is at equilibrium. Let's do a practice problem. Human blood naturally has a concentration of 0.09% salt. If a physician administers an IV saline drip of 0.25% into the bloodstream of a patient, which of the following is most likely to occur? Could it be that there is no change? Red blood cells will maintain homeostasis. The red blood cells will swell and lyse as they try to work to maintain osmolarity. Or will the red blood cells shrivel and become non-functional? What do you think? Well, in this case, those red blood cells are going to shrink and shrivel. Why? This is a hypertonic solution for the blood cells. The blood cells have a concentration of 0.09% salt, but the solution they're in has a much greater concentration of solute at 0.25. Because of that, osmosis always moves from a higher concentration to a lower concentration, so water will exit the red blood cells and go to the solution. Here are some tips when doing osmosis problems. Generally speaking, in most of these problems, water is the only substance that can move across the membrane unless the problem tells you otherwise. If the problem tells you another substance is permeable, then keep that in mind and anticipate the movement of those. For example, here I have a cell that has glucose and starch in a solution that is glucose and starch. Let's say the cell in the solution to the right is permeable to glucose, but is not permeable to starch. You might be asked to predict the movement of water, but keep in mind, since it's permeable to glucose, this glucose will move as well. So you want to anticipate that glucose movement to equilibrium in addition to the movement of water. Another thing you can encounter with osmolarity problems is pulling information from a graph. For example, this is a famous experiment where you take potatoes, slice cores out of them, and soak them in different concentrations of sugar. So here you're seeing the change in potato mass after they're being soaked and the concentration of sugar they're soaked in. If given a graph like this, you could be asked, what molarities is the potato core in a hypertonic solution? To solve this, you want to pull on your knowledge of solutions. Remember that a hypertonic solution is a solution when water is going to be exiting the cell. So if water is exiting a potato, that means the potato is going to be losing mass. So the answer to this would be between 0.3 molar and 1 molar. And this question could be asked in other ways. What solution is the potato hypotonic? Well, with hypotonic, the cells gain water, so the potato is going to gain mass. At what solution is it isotonic? Well, at the point where they have no change in mass, same amount of water out as the same amount of water in. So let's get mathematical. Osmolarity problems can be solved as we did before by assessing the concentrations outside and inside the cell, but we can get more specific by measuring a system's water potential. The water potential is a measure of a system's potential energy, meaning we're quantifying the tendency of water to move. We're able to use math to determine how osmosis will occur. Here's an example of how we can do that. The equation of water potential is bar equals bar pressure plus bar solute potential. We'll explain what those mean in a moment, but I just want you to keep in mind the big picture here is we're just using math to determine what direction water will move according to osmosis. A big thing to keep in mind is this is on your AP Biology equation sheet, meaning you'll have full access to the equation during the exam, and problems like this are fair game on your AP test. So water potential, or bar, is determined by two factors. The first one is the pressure potential. This is just a measure of the pressure that's being put on a system. If there is no pressure being put on the system, like right here, it's just an open container, the pressure potential is 0. But if pressure is being applied physically by an apparatus like in this picture, or by a cell membrane or a cell wall, the pressure potential value can be positive. So for this apparatus right here, I would say the pressure potential is 0 on the left-hand side because it's an open container, it is open atmosphere, but here pressure is being applied, and I happen to know that it's a pressure potential of 1. The second component of water potential is the solute potential. This is a measure of how much solute is in the solution. If there is no solute in the solution, if it's a situation like distilled or pure water, the solute potential is a value of 0. If there is solute in the solution, it can have incrementally negative values. So to refer to this apparatus again, on the left-hand side where I have pure water, that is the solute potential of 0. There's no solute in the solution. This solution, however, has quite a bit of solute and has a solute potential of -1. So plugging these values into the water potential equation, I add the pressure to the solute, and that gives me a value of a water potential of 0 for both sides. But what do these values mean? Well, it depends on the situation you're working with. Let's look at a couple examples. As a general rule of thumb when working with water potential problems, water will always move to the most negative water potential. Or to say another way, osmosis will move to the most negative bar. So in this example here, which is similar to the last problem, but there's no pressure, on the left-hand side I have a water potential of 0, and on the right-hand side a water potential of -1. What happens in this system is water moves to the right-hand side. There will be a net movement of water to where there is a negative water potential until the system reaches equilibrium. Here is identical to the previous problem. If I apply pressure to the right-hand side, however, that will move that negative value to being 0. With a water potential of 0 on both sides, there is net no movement of water at all. And yes, you can do this in the opposite direction. I could apply even more pressure on the right-hand side to get the water to move to the left. This is how water filtration systems like reverse osmosis work. By applying pressure to water, we can force water out of heavy solute solutions. Think desalination, getting fresh water out of the ocean. Here's an example that's applicable to cells. If I put this plant cell in a pure water solution with a water potential of 0, but the water potential inside the cell is -1, well, water's going to rush in. Do you remember what kind of solution that is? That's a hypotonic solution. Let's say, though, the cell is turgid, meaning firm, or it has more water inside of it, so it's applying more pressure. If I have a water potential of 0 on both sides, well, I'm going to have a net no movement of water. Now, that's a simplified version of water potential. Solute potential, the bar S, actually is usually not provided to you. You have to calculate it using the equation bar S equals -ICRT. That's a lot of variables. Take a breath. This equation is on your AP biology equation sheet. You will always have access to it during your test. Let's break down what each letter means. The lowercase I stands for the ionization constant. This typically is provided to you in the problem and may even be provided on the equation sheet. The equation sheet lets you know that if you have a sugar solution, the ionization constant is 1. C stands for the molar concentration. This is something you have to get from the experimental data provided to you. It's asking you what is the concentration of solute in moles for either in the cell or the solution, whichever system you're calculating. If you're reading this off of a graph, that is that isotonic point that I mentioned to you earlier. You're looking for the point where there's no change in mass in the solution. That is where you find the molar concentration, or C value, when calculating the solute potential. R is a constant, and it's given to you on the equation sheet. It is the pressure constant, and it is always 0.0831 liter bar per mole K. It's always there, just plug it in. And then T is the temperature. For this equation, the temperature needs to be provided in Kelvin. So if you're given a temperature in Celsius, just add 273 to get the Kelvin value. Let's do a practice problem for water potential. The molar concentration of a sugar solution in an open beaker has been determined to be 0.3 molar. Calculate the solute potential at 27 degrees Celsius. Round your answer to the nearest hundredth. What is the water potential? Well, I know my water potential equation is bar equals bar P plus bar S. So let's first figure out bar P, the pressure potential. The problem tells me it's in an open beaker, and my equation sheet tells me it is a pressure potential of 0 if it is an open solution or container. So now I know water potential equals 0 plus the solute potential. Let's find the solute potential. The solute potential equation is -ICRT, so now we just need to find each value and plug it in. To find -I, that is the ionization constant. The problem tells me this is a sugar solution, so according to the equation sheet, the value for a sugar solution is 1. Now we need to find C, which is the molar concentration. This is, again, provided in the problem. It's already been determined the molar concentration is 0.3 molar. So go ahead and plug that in. Now we have R. R is the pressure constant, so we just write the pressure constant 0.0831 liter bars per mole per Kelvin. Now for the temperature does need to be in Kelvin. We're provided 27 degrees Celsius, so we add that to 273 and get 300 Kelvin. So to find bar S, we just multiply all the values together, and please do not forget that negative sign on the outside. I've seen some heartbreaking instances of students leaving that out. The equation is negative ICRT, and that gives us a solute potential of negative 7.48 bar. Plug that into our water potential. Now we have 0 plus negative 7.48 for a water potential of negative 7.48. So going back to our tale of blood and water, what was happening in these situations? It was osmotic pressure. Some instances, the solution was isotonic, like you saw in the beginning. In the second situation, when the cells began to shrivel, you saw a hypertonic solution. And in the last situation, when the cells lysed, you saw a hypotonic solution. But why is all this water moving? Well, osmosis, and we can use water potential to calculate it. Now you have the tools to be able to demonstrate why water would be moving inside or out of a cell. I hope this lecture was helpful in understanding osmosis and how to solve water potential problems, and I will see you next time.